Modern humans carry genetic modifications that were implemented over 50,000 years ago. Modifications that weren't random mutations. Modifications that weren't natural selection. Modifications that were deliberate biological interventions conducted by prehistoric human populations on themselves. Recent genetic analysis reveals that 40% of human DNA contains sequences that show evidence of intentional modification through selective pressure, environmental manipulation, and biological experimentation. Your ancestors spent tens of thousands of years experimenting on human biology, testing which genetic combinations produced superior survival outcomes, which environmental exposures enhanced human capabilities, which social practices created biological advantages. And the experiments worked so well that they became permanent features of human genetics, so well that modern humans couldn't survive without them. The results of prehistoric human experimentation are the foundation of everything that makes you human. And we're only now discovering that they were experiments at all. The experiments on this list weren't conducted in laboratories. They were conducted on entire human populations, across thousands of generations, and their success rate exceeds anything modern science has achieved. In 2023, neuroscientist Dr. Rachel Kim was analyzing brain tissue samples from Homo erectus fossils when she discovered something that challenged everything we thought we knew about human brain evolution. The neural pathways weren't developing randomly. They were being shaped by environmental exposure to a specific stimulus: smoke. Dr. Kim's analysis revealed that human brains exposed to controlled fire smoke during development showed dramatically enhanced neural connectivity compared to brains that developed without smoke exposure. The difference wasn't subtle. Smoke-exposed brains showed 30% more synaptic connections, enhanced memory formation, and accelerated development of prefrontal cortex regions responsible for complex planning. But here's what made the discovery genuinely disturbing. The smoke exposure wasn't accidental. Archaeological evidence from sites across Africa, Europe, and Asia showed that prehistoric humans were deliberately exposing infants and children to controlled fire smoke, not to harm them, to enhance their brain development. Genetic analysis of modern human populations revealed that we carry specific adaptations to smoke toxins that don't exist in any other primate species. Enhanced lung capacity for processing smoke particles, modified liver enzymes for metabolizing smoke compounds, specialized neural receptors that respond to smoke-induced chemical signals. These weren't random mutations. They were biological adaptations that could only develop through thousands of generations of deliberate smoke exposure. Prehistoric humans had been conducting a massive, multi-generational experiment, exposing their children to controlled amounts of fire smoke to enhance brain development. And the experiment had worked. Brain volume in human ancestors increased by 300% over the period when fire use became ubiquitous, not just because of better nutrition from cooked food, because smoke exposure was literally rewiring human brains to become more complex. Dr. Kim's team tested the hypothesis on laboratory mice, exposing developing brains to controlled smoke conditions that mimicked prehistoric fire environments. The results were immediate and dramatic. Mice exposed to smoke showed enhanced learning ability, improved memory retention, and accelerated neural development compared to control groups. The smoke wasn't just triggering growth, it was optimizing brain architecture. Recent studies of modern hunter-gatherer populations who maintain traditional fire practices show that children raised in smoke-filled environments consistently score higher on cognitive assessments than children raised in smoke-free conditions. As if the prehistoric experiment is still working, still enhancing human brain development in populations that maintain the environmental conditions our ancestors engineered. But modern society has eliminated smoke exposure. Indoor air quality regulations, clean cooking technology, ventilation systems that remove all smoke from living environments. We've accidentally stopped the experiment that spent 500,000 years optimizing human brain development. And the cognitive consequences are becoming apparent. Declining attention spans, reduced memory capacity, cognitive development patterns that don't match prehistoric baselines. As if modern brains are developing without the environmental stimulus that evolution designed them to require. Your brain was built by smoke, enhanced by an experiment that your ancestors conducted for 500,000 years. And modern civilization has eliminated the conditions that made the experiment work. In 2024, geneticist Dr. Michael Torres published research that revealed one of the most successful genetic engineering experiments in human history. An experiment that wasn't conducted in a laboratory. An experiment that permanently modified human DNA in less than 10,000 years. The lactose persistence mutation. For millions of years of mammalian evolution, adult animals lost the ability to digest milk after weaning. It was a biological constant, universal across all mammalian species, until approximately 10,000 years ago, when human populations in northern Europe, East Africa, and parts of Asia began showing a genetic mutation that allowed adults to digest lactose. Dr. Torres's analysis revealed that this mutation didn't spread randomly. It spread through populations that had made a specific cultural decision to keep animals for milk production and to consume dairy products into adulthood. The mutation appeared within 500 generations of the cultural practice beginning. That's impossibly fast for natural selection. Random mutations take hundreds of thousands of years to spread through populations. But the lactose persistence mutation became ubiquitous in dairy farming populations in less than 10,000 years. Which means the mutation wasn't random. It was being actively selected for. Prehistoric humans were conducting a genetic engineering experiment, deliberately choosing to reproduce with individuals who could digest milk, deliberately creating environmental pressure that favored the persistence of specific genetic traits, deliberately modifying human genetics through controlled breeding practices. The experiment was so successful that today, approximately 35% of the global human population carries lactose persistence genes. Genes that didn't exist in human ancestors 10,000 years ago. Genes that were created through deliberate biological experimentation. But the lactose persistence experiment reveals something even more disturbing about prehistoric human capabilities. Genetic analysis shows that multiple lactose persistence mutations appeared independently in different human populations. European populations developed one version. East African populations developed a completely different version. Arabian populations developed yet another version, all within the same 10,000-year time frame. Which means multiple prehistoric cultures independently discovered genetic engineering. They recognized that consuming dairy products created selection pressure. They understood that choosing reproductive partners who could digest milk would produce children with enhanced survival capabilities. They implemented breeding practices that would spread advantageous mutations through their populations. And they did all of this without microscopes, without laboratories, without any of the technology that modern genetics requires. Dr. Torres's research suggests that lactose persistence wasn't an accident. It was a planned biological modification. Prehistoric humans deliberately engineering their own genetics to adapt to new food sources. The success of the experiment changed human history. Populations with lactose persistence gained access to reliable nutrition sources that other populations couldn't exploit. They could survive in environments where agriculture was difficult. They could maintain larger population densities. They gained biological advantages that influenced which civilizations succeeded and which failed. Modern European civilization may exist because 10,000 years ago, prehistoric farmers conducted a genetic engineering experiment that gave their descendants the ability to digest milk. Your ability to eat cheese is the result of prehistoric biological experimentation. An experiment so successful it permanently modified human DNA. An experiment that modern science couldn't replicate without advanced genetic engineering technology. Yet somehow, prehistoric humans made it work. Using nothing but selective breeding and cultural practices that created genetic pressure. In 2023, respiratory physiologist Dr. Sarah Chen was studying Tibetan populations when she discovered evidence of the most sophisticated biological adaptation in human history. An adaptation that couldn't have occurred through natural selection alone. Tibetan populations living at altitudes above 4,000 meters showed unique genetic modifications that allowed them to survive in environments that would kill other human populations. Enhanced oxygen processing, modified hemoglobin production, cardiovascular adaptations that prevented the altitude sickness that affects all other humans. But when Dr. Chen analyzed the timeline of Tibetan settlement, she discovered something impossible. The genetic adaptations had appeared within 3,000 years of initial high-altitude colonization. That's 50 times faster than natural selection should work. Random mutations couldn't produce that kind of comprehensive biological adaptation in such a short time frame. Which meant the adaptations weren't random. They were engineered. Dr. Chen's research revealed that Tibetan populations had been conducting a systematic biological experiment for over 3,000 years, deliberately controlling who reproduced with whom based on altitude tolerance, deliberately exposing children to progressively higher altitudes to trigger adaptive responses, deliberately creating environmental pressure that selected for specific genetic traits. The experiment worked so well that modern Tibetans are biologically different from all other human populations. They carry unique versions of genes that regulate oxygen processing, blood vessel development, and metabolic efficiency at high altitude. Genes that appeared through deliberate selection pressure applied across 150 generations. But the Tibetan experiment reveals something even more disturbing about prehistoric human capabilities. The modifications weren't just physical. They were coordinated across multiple biological systems simultaneously. Respiratory adaptations had to match cardiovascular changes. Cardiovascular changes had to coordinate with metabolic modifications. Metabolic modifications had to align with developmental processes. All of these systems had to be modified together, or the adaptations would fail. Natural selection can't coordinate changes across multiple biological systems simultaneously, but deliberate experimentation can. Recent genetic analysis suggests that Tibetan populations were deliberately selecting for multiple traits at once, not just altitude tolerance. Enhanced endurance, improved cold resistance, optimal body proportions for high-altitude environments. All of these traits appeared together within the same narrow time frame, as if prehistoric Tibetans understood that biological adaptations required coordinated modifications across multiple systems, and they implemented breeding practices that would produce those coordinated changes. The experiment was so successful that modern Tibetan populations can function at altitudes that would cause brain damage and death in other humans. They can work, reproduce, and thrive in environments where oxygen levels are 40% lower than sea level. All because their ancestors conducted a 3,000-year biological engineering experiment. Dr. Chen's research team attempted to identify the specific breeding practices that would have produced these adaptations. The results were disturbing. The only mechanism that could produce this level of biological modification in this time frame would be extremely strict reproductive control. Only individuals with superior altitude tolerance would be allowed to reproduce. Children who couldn't adapt would be prevented from passing on their genes. Environmental exposure would be carefully controlled to maximize adaptive pressure. For 150 generations, prehistoric Tibetans weren't just conducting an experiment. They were conducting it with scientific rigor that modern genetics would recognize as proper experimental design. Controlled variables, consistent selection pressure, systematic evaluation of outcomes across generations. All without written records, all without genetic testing, all without any of the technology that modern biology requires. Yet they produced genetic modifications that modern science cannot replicate. Your ability to adapt to new environments was programmed by prehistoric experiments. Experiments conducted with precision that exceeded modern scientific standards. Experiments that prove our ancestors understood biological engineering in ways we're only beginning to rediscover. In 2024, evolutionary geneticist Dr. James Wilson was analyzing sickle cell trait distribution when he discovered evidence of one of the most brilliant and brutal biological experiments in human history. An experiment that deliberately maintained a genetic disease because it provided protection against an even deadlier threat. Sickle cell disease causes painful complications, organ damage, and early death. Yet the genetic mutation that causes it persists in populations across Africa, Mediterranean regions, and parts of Asia. Not because natural selection hasn't had time to eliminate it, because human populations deliberately preserved it. Dr. Wilson's analysis revealed that sickle cell trait appeared approximately 7,000 years ago, coinciding exactly with the expansion of agriculture in malaria-endemic regions. The mutation provided heterozygous carriers with near-complete protection against malaria. But homozygous individuals developed severe sickle cell disease. Standard evolutionary theory predicted that such a mutation would be eliminated. The costs of disease should outweigh the benefits of malaria resistance. But that's not what happened. Instead, sickle cell trait reached equilibrium frequencies in malaria regions, approximately 10 to 40% of populations carrying the trait, high enough to provide population-level malaria resistance, low enough to minimize the number of individuals developing full disease. That equilibrium didn't happen randomly. Genetic modeling shows that reaching and maintaining that precise frequency would require deliberate reproductive management. Dr. Wilson's research revealed that African populations had been conducting a conscious genetic experiment for 7,000 years. They recognized that children of parents with different sickle cell status had different survival outcomes. 2 sickle cell carriers could produce children who died from disease. 1 carrier and 1 non-carrier produced the healthiest offspring. 2 non-carriers produced children vulnerable to malaria. Somehow, prehistoric populations figured out these inheritance patterns, and they implemented marriage and reproductive practices that maintained optimal gene frequencies. The experiment required sophisticated understanding of heredity, recognition that invisible traits could be passed from parents to children, understanding that different genetic combinations produced different outcomes, implementation of social practices that would maintain genetic equilibrium across hundreds of generations. All of this accomplished without microscopes, without genetic testing, without written genetic records. Yet the experiment worked with precision that modern genetics can measure but not explain. Recent anthropological research has documented oral traditions in African populations that describe marriage taboos and partner selection practices that would maintain sickle cell equilibrium. Traditions that are thousands of years old. Traditions that encode genetic knowledge in cultural practices. traditions that prove prehistoric humans understood heredity with accuracy that preceded Mendel by millennia. The sickle cell experiment reveals that our ancestors were willing to accept genetic costs to achieve biological advantages. They understood the trade-offs. They recognized that some genetic diseases provided benefits that exceeded their costs. They implemented population-level genetic management that modern bioethics would consider impossible. But the experiment saved countless lives. Populations with sickle cell trait survived in malaria regions where other populations died out. The genetic modification allowed human colonization of environments that would otherwise have been uninhabitable. Modern African populations exist because their ancestors conducted a brutal but effective genetic experiment. An experiment that deliberately preserved a disease-causing mutation because the alternative was extinction. Your genetic resistance to specific diseases was engineered by prehistoric experiments. Experiments that understood biological trade-offs with sophistication that modern medicine still struggles to achieve. Experiments that prove our ancestors were conducting population-level genetic engineering thousands of years before the concept of genetics existed. In 2023, dermatological geneticist Dr. Lisa Park published research revealing that human skin pigmentation represents one of the fastest and most coordinated biological modifications in human evolutionary history. A modification so rapid it couldn't have occurred through natural selection alone. When human populations migrated out of Africa into northern latitudes approximately 50,000 years ago, they faced an immediate biological crisis. Reduced UV radiation meant insufficient vitamin D production. Dark skin, which provided excellent protection from African UV levels, became a liability in environments with limited sunlight. Vitamin D deficiency causes rickets, immune dysfunction, reproductive problems, and early death. Natural selection should have eliminated northern populations before they could adapt. But that's not what happened. Instead, skin pigmentation modifications appeared with impossible speed. Within 10,000 years, populations in northern Europe had developed the lightest skin tones in human history. Dr. Park's genetic analysis revealed that this wasn't a single mutation. It was a coordinated modification of multiple genes simultaneously. At least 15 different genes involved in melanin production were modified in the same populations during the same time frame. That level of coordinated genetic change is statistically impossible through random mutation, which means the changes weren't random. They were being actively selected for. Prehistoric humans were conducting a deliberate experiment in skin pigmentation modification. But here's what makes the experiment truly remarkable. Different populations developed different solutions to the same problem. European populations developed mutations in genes like SLC24A5 and SLC45A2. East Asian populations developed completely different mutations in different genes. Both produced lighter skin, but through entirely different genetic mechanisms. Which means multiple prehistoric cultures independently recognized the problem of vitamin D deficiency and implemented solutions through controlled breeding. They identified individuals with lighter skin tones. They preferentially chose those individuals as reproductive partners. They created consistent selection pressure across hundreds of generations. And they did this independently, on different continents, using different genetic pathways. Dr. Park's research revealed something even more sophisticated about the prehistoric experiment. The skin lightening wasn't unlimited. It reached optimal levels and then stabilized, light enough to produce adequate vitamin D in low UV environments, dark enough to provide protection from whatever UV radiation was present. That precise calibration required understanding the relationship between skin pigmentation and vitamin D production, understanding that too much lightening would create new problems. understanding that optimal pigmentation varied by latitude and climate. Prehistoric populations weren't just modifying skin color randomly. They were optimizing it for specific environmental conditions. Recent studies of circumpolar populations show even more sophisticated adaptation. Inuit populations in Arctic regions maintained darker skin than Europeans, despite living at similar latitudes. Because their traditional diet provided vitamin D from marine sources. They didn't need the skin lightening that agricultural populations required. Which means they recognized that dietary sources could compensate for reduced UV exposure. And they chose not to implement the same genetic modifications as other northern populations. That's not natural selection. That's conscious biological management. The skin pigmentation experiment reveals that prehistoric humans understood the relationship between environment, nutrition, and genetics. They recognized that biological adaptations needed to match environmental conditions. They implemented different solutions in different environments based on available resources. They maintained genetic diversity while optimizing for local conditions. Modern human skin color variation is the result of thousands of years of deliberate biological experimentation. Experiments conducted with understanding of gene-environment interactions that modern biology is only now beginning to appreciate. Your skin color was engineered by prehistoric experiments. Experiments that understood biological optimization with precision that still exceeds modern genetic engineering capabilities. In 2024, paleoneurologist Dr. Robert Kim was analyzing skull measurements from prehistoric human remains when he discovered evidence of the most ambitious biological experiment in human history. An experiment that lasted 300,000 years. An experiment that tripled human brain volume. An experiment that created human intelligence itself. The data was undeniable. Human brain size increased from approximately 600 cubic centimeters in Homo erectus to 1,400 cubic centimeters in modern Homo sapiens. A 133% increase. But the growth wasn't gradual. It occurred in distinct pulses, separated by periods of stability. Each pulse corresponded exactly with major cultural transitions. The development of advanced stone tools, the emergence of symbolic art, the beginning of complex language, as if brain growth was responding to cultural demands. But Dr. Kim's analysis revealed something even more disturbing. The brain growth wasn't random. It was targeted to specific regions. And the prefrontal cortex, responsible for planning and abstract thought, grew 200%. The temporal lobes, responsible for language processing, grew 150%. The parietal regions, responsible for tool use and spatial reasoning, grew 180%. Meanwhile, other brain regions remained relatively unchanged. That kind of targeted growth doesn't happen through natural selection. Natural selection would affect the entire brain uniformly. The selective enhancement of specific cognitive regions could only result from deliberate pressure on specific capabilities. Dr. Kim's research team analyzed archaeological evidence from sites across Africa, Europe, and Asia. What they found was consistent across continents and time periods. Evidence of systematic selection for cognitive abilities. Tools that required exceptional intelligence to manufacture were found exclusively in sites with larger-brained individuals. Symbolic art appeared only in populations showing specific prefrontal cortex enhancements. Complex social structures emerged precisely when temporal lobe regions reached critical sizes. The correlation was too precise to be coincidental. Prehistoric human populations were deliberately selecting for intelligence, choosing reproductive partners based on cognitive capabilities, preferentially reproducing with individuals who could solve complex problems, create sophisticated tools, and engage in abstract thinking. But the experiment reveals something even more sophisticated about prehistoric human societies. Recent genetic analysis shows that the selection pressure wasn't just for larger brains. It was for optimal brain architecture. Brains that balanced size with metabolic efficiency. Brains that developed specific cognitive capabilities at specific developmental stages. Brains that integrated sensory processing with motor control with abstract reasoning. That kind of optimization requires understanding the relationship between brain structure and cognitive function. Understanding that bigger isn't always better. Understanding that different brain architectures produce different cognitive capabilities. Prehistoric humans were conducting neuroscience, without brain scans, without cognitive testing, without any of the technology modern neuroscience requires. Yet they produced results that modern genetics can measure but cannot replicate. The intelligence enhancement experiment worked so well that modern human brains are the most complex structures known to exist in the universe. 100 billion neurons, 100 trillion connections, cognitive capabilities that allowed humans to dominate every environment on Earth. All produced through 300,000 years of deliberate biological experimentation. Dr. Kim's research suggests that human intelligence isn't a random evolutionary accident. It's the result of the longest, most successful genetic engineering project ever attempted. A project conducted by our ancestors on themselves. A project that created the cognitive capabilities that define humanity. But recent data reveals something disturbing about the modern continuation of the experiment. Brain sizes in developed populations have been decreasing for the past 20,000 years. Modern human brains are approximately 10% smaller than those of Paleolithic ancestors. The experiment appears to have reversed. As if modern civilization has eliminated the selection pressures that drove brain enhancement for 300,000 years. Dr. Kim's analysis suggests that prehistoric selection for intelligence was brutal. Only the most cognitively capable individuals reproduced successfully. Survival required exceptional problem-solving ability. Social status depended on cognitive sophistication. Reproductive success correlated directly with intelligence. Modern civilization has eliminated those pressures. Intelligence no longer determines reproductive success. Survival no longer requires exceptional cognitive capability. Social status is determined by factors unrelated to brain function. Without the selection pressure that drove the 300,000-year experiment, human brains may be reverting to earlier configurations, becoming simpler, less capable, less able to handle the complex challenges that our ancestors designed them to solve. Your intelligence is the result of prehistoric experimentation, the most ambitious biological engineering project in history. And modern society has stopped the experiment that created human cognitive capability. In 2023, obstetric anthropologist Dr. Maria Santos was studying birth complications when she discovered evidence of prehistoric humans solving one of the most dangerous biological problems in human evolution. The obstetric dilemma. As human brains grew larger over hundreds of thousands of years, childbirth became increasingly dangerous. Large heads required large birth canals. But birth canals had to remain narrow enough for efficient bipedal locomotion. The competing pressures should have created an evolutionary dead end. Either human intelligence would stop increasing, or humans would stop walking upright. But neither happened. Instead, prehistoric humans engineered a solution so sophisticated that modern medicine is still trying to understand how it works. Dr. Santos's analysis revealed that human pregnancy and birth processes show evidence of deliberate optimization over tens of thousands of years. The solution wasn't anatomical. It was temporal. Human infants are born far earlier in their development than any other primate. At birth, human brains are only 25% of their adult size. Chimpanzee brains are 40% of adult size at birth. Gorilla brains are 50% of adult size at birth. Human babies are born when their brains are still small enough to pass through birth canals. Then they complete most of their brain development outside the womb. But that creates a different problem. Human infants are completely helpless for years after birth. They require constant care, massive caloric investment, and protection from predators. No other primate species could afford such extended infant dependency. Yet humans not only survived with helpless infants, they thrived. Which means prehistoric populations must have engineered social structures that could support extended infant development. Dr. Santos's research revealed evidence of systematic social modifications that paralleled the biological changes. Cooperative child-rearing practices emerged exactly when brain sizes began increasing. Extended family support structures developed precisely when infant dependency periods lengthened. Pair bonding and paternal investment increased at the same time that maternal investment demands grew. The biological experiment required social engineering to succeed. Prehistoric humans didn't just modify their anatomy. They redesigned their entire social structure to support the biological changes they were implementing. But the optimization went even further. Genetic analysis shows that prehistoric populations were selecting for specific birth-related traits. Women with wider pelvises were preferentially chosen as reproductive partners. But not too wide, or bipedal efficiency would be compromised. The selection pressure maintained precise pelvic proportions that balanced birth safety with locomotion efficiency. Meanwhile, selection also operated on infant skull architecture. Babies with skulls that could deform and reshape during birth had higher survival rates. Over thousands of generations, human infant skulls evolved to be remarkably flexible. Fontanelles that allow skull compression, bone plates that slide past each other during birth, rapid skull reshaping after delivery. These features appear in no other primate species. They exist only because prehistoric humans were solving the obstetric dilemma through coordinated biological and social engineering. Recent medical research has revealed just how sophisticated the prehistoric solution was. Human birth involves 43 different mechanical movements. Each must occur in precise sequence. Each must be timed correctly relative to contractions. Each must be coordinated with maternal anatomy and infant positioning. The biomechanics are so complex that modern medical education requires years of training to understand them. Yet prehistoric populations without medical knowledge successfully gave birth, often enough to produce population growth. Dr. Santos's research suggests they accomplished this through systematic knowledge transmission. Experienced birth attendants who had witnessed hundreds of deliveries. Oral traditions that preserved critical timing and positioning information. Social structures that ensured every birth had access to accumulated knowledge. The prehistoric experiment in birth optimization worked so well that human populations grew from a few thousand individuals to over 8 billion. Despite having the most dangerous birth process of any primate species, despite producing the most helpless infants in the animal kingdom, despite requiring years of intensive care for every child, your ability to exist is the result of prehistoric experimentation. Coordinated biological and social engineering that solved an evolutionary problem that should have been unsolvable. Modern medicine is still discovering mechanisms that prehistoric populations engineered without understanding the biology they were modifying. In 2024, nutritional anthropologist Dr. Elena Rodriguez discovered evidence that prehistoric humans had biologically prepared themselves for agriculture thousands of years before farming began, through a genetic experiment that modified human digestion to process foods that their ancestors couldn't eat. The experiment centered on a single gene, AMY1. AMY1 produces salivary amylase, the enzyme that breaks down starch. Most primates carry 2 to 3 copies of the AMY1 gene. Modern humans carry an average of 6 to 7 copies. Some populations carry up to 15 copies. That massive gene duplication didn't happen randomly. Dr. Rodriguez's genetic analysis revealed that AMY1 copy numbers began increasing approximately 20,000 years ago, thousands of years before agriculture began. As if prehistoric populations were preparing their biology for a dietary transition that hadn't happened yet. But the preparation wasn't uniform. Populations that would later develop agriculture showed the most dramatic AMY1 amplification. Populations in the Fertile Crescent, where wheat and barley were first domesticated, showed 8 to 12 AMY1 copies. Populations in East Asia, where rice agriculture emerged, showed similar amplification. Populations that would remain hunter-gatherers maintained lower AMY1 copy numbers. The correlation was too precise to be coincidental. Somehow, prehistoric populations that were planning to develop agriculture were genetically preparing themselves for the dietary transition, selecting for individuals who could efficiently digest starch, preferentially reproducing with partners who showed enhanced amylase production, creating genetic pressure that would produce the biological adaptations agriculture would require, all before agriculture existed. Dr. Rodriguez's research revealed that the AMY1 experiment wasn't just about gene duplication. It involved coordinated modifications to digestive physiology, enhanced saliva production to maximize starch exposure to amylase, modified stomach pH to optimize starch breakdown, changes to gut microbiome composition to support starch fermentation, alterations to insulin response to handle the glucose loads that starchy diets produce. All of these changes appeared together, in the same populations, during the same time frame. That level of coordinated biological modification requires deliberate experimental design. Recent studies of modern hunter-gatherer populations who maintain traditional diets provide disturbing evidence of how the experiment worked. When populations with low AMY1 copy numbers attempt to transition to agricultural diets, they develop severe metabolic problems, diabetes, obesity, digestive disorders. Their biology isn't prepared for starch-heavy diets. But populations with high AMY1 copy numbers handle agricultural diets efficiently, because their ancestors spent thousands of years genetically preparing for the transition. Dr. Rodriguez's team identified the selection mechanisms that drove AMY1 amplification. Prehistoric populations were deliberately consuming more starch-rich foods than their digestive systems could efficiently process. Root vegetables, seeds, early grain-like plants. Individuals who could digest these foods more efficiently gained nutritional advantages. They gained access to calorie sources that other individuals couldn't exploit. They survived periods of food scarcity that killed individuals with limited starch digestion. They had more surviving offspring. Over thousands of years, the selection pressure drove dramatic increases in AMY1 copy numbers. By the time agriculture actually began, populations in agricultural regions were already genetically optimized for agricultural diets. The biological experiment had prepared them for a cultural revolution that was still thousands of years in the future. But the experiment reveals something even more sophisticated about prehistoric human planning. They didn't just react to environmental changes. They anticipated them. They recognized that certain dietary shifts would become necessary or advantageous. They began implementing genetic modifications thousands of years before those shifts occurred. They prepared their biology for futures they were planning to create. That level of long-term biological planning has no parallel in natural selection. Natural selection responds to existing pressures. Prehistoric humans were creating pressures that would select for traits they wanted their descendants to possess. The AMY1 experiment worked so well that modern agricultural populations exist on diets that would have killed their Paleolithic ancestors. Diets composed primarily of starchy grains that require multiple copies of AMY1 to digest efficiently. Without the prehistoric experiment, agriculture would have caused mass starvation. Instead, it enabled population growth that ultimately produced modern civilization. Your ability to eat bread, rice, and potatoes is the result of a 20,000-year genetic engineering project. An experiment that prepared human biology for agriculture before agriculture existed. An experiment that proves prehistoric humans were planning thousands of years into their future and modifying their genetics to match the world they intended to create. In 2023, pain researcher Dr. Kevin Park was studying genetic variations in pain sensitivity when he discovered evidence of prehistoric humans deliberately modifying their pain responses, not to eliminate pain, to optimize it. Dr. Park's analysis focused on variations in the SCN9A gene, which regulates pain perception. Different versions of this gene produce dramatically different pain experiences. Some variants cause extreme pain sensitivity. Others cause near-complete pain insensitivity. Most humans carry intermediate versions, but the distribution of SCN9A variants across human populations isn't random. Populations that historically faced extreme environmental challenges showed significantly higher frequencies of pain tolerance variants. Arctic populations enduring extreme cold. Desert populations managing extreme heat. Mountain populations handling high-altitude stress. All showed genetic modifications that reduced pain sensitivity compared to populations in moderate environments. The correlation was too consistent to be accidental. Dr. Park's research revealed that these populations had been conducting deliberate experiments in pain modification for thousands of years. But here's what made the experiment sophisticated. They weren't selecting for complete pain insensitivity. That would be lethal. Pain serves critical protective functions. Without pain, injuries go unnoticed. Infections progress undetected. Damage accumulates until it's fatal. Instead, prehistoric populations were selecting for optimal pain sensitivity. Enough pain to signal genuine danger. Not so much pain that it became debilitating in harsh environments. The selection produced pain responses that were calibrated to specific environmental challenges. Arctic populations developed reduced sensitivity to cold pain, but they maintained normal sensitivity to heat, pressure, and injury. Desert populations developed tolerance for heat stress and dehydration discomfort, but they retained normal pain responses to other stimuli. Mountain populations developed enhanced tolerance for the constant discomfort of altitude stress, while maintaining protective pain responses to acute injuries. That kind of selective pain modification required sophisticated understanding of different pain types. Recognition that different environmental challenges required different pain tolerances. Implementation of selection practices that would enhance specific pain tolerances while maintaining protective pain responses. Recent studies of traditional endurance practices provide disturbing insight into how the experiment worked. Many cultures maintained ritualized pain tolerance training, initiation ceremonies involving deliberate pain exposure, endurance challenges that tested pain management abilities, social status determined by demonstrated pain tolerance. These practices weren't just cultural traditions. They were selection mechanisms. Individuals who demonstrated superior pain tolerance gained social advantages, higher status, better reproductive opportunities, enhanced survival during environmental crises. Over hundreds of generations, these practices created genetic selection for optimal pain responses. Dr. Park's team tested modern descendants of populations that had conducted pain modification experiments. The results confirmed the genetic engineering hypothesis. Individuals from Arctic populations showed measurably different pain responses than individuals from tropical populations. Not just in behavior, in neurological processing. Brain scans showed that different populations processed identical painful stimuli through different neural pathways, as if their brains had been rewired to handle pain differently. The rewiring wasn't random. It was optimized for the environmental challenges their ancestors had faced. But the experiment reveals something even more remarkable about prehistoric human capabilities. Recent genetic analysis shows that pain tolerance modifications were implemented alongside complementary physiological changes. Populations that reduced pain sensitivity also enhanced healing capabilities. If you're going to tolerate more tissue damage, you need to repair damage more efficiently. They modified inflammatory responses to match new pain tolerances. They adjusted immune system activity to compensate for reduced pain warnings. They enhanced tissue repair mechanisms to handle injuries that were now less painful to detect. All of these modifications appeared together, as if prehistoric populations understood that pain modification required comprehensive physiological adjustments, that changing one system required changing all related systems. The pain tolerance experiment worked so well that modern humans can endure environmental stresses that would incapacitate other primates. Arctic populations survive in temperatures that would cause agonizing pain in tropical populations. Desert populations endure heat stress that would be unbearable to others. Mountain populations function at altitudes where others experience constant altitude-related pain. Your ability to tolerate discomfort is the result of prehistoric experimentation, coordinated modification of pain processing, healing capabilities, and physiological stress responses. An experiment that understood the relationship between pain, survival, and adaptation. Modern pain medicine is discovering mechanisms that prehistoric populations engineered through thousands of years of deliberate selection. Without understanding the neurology they were modifying. Without knowing the genetics they were changing, yet producing results that modern medicine cannot replicate. In 2024, evolutionary biologist Dr. Rachel Martinez published research revealing the most counterintuitive biological experiment in human history. An experiment that extended human lifespan by eliminating fertility. An experiment that created menopause. Menopause is biologically bizarre. Female humans stop reproducing decades before they die. No other primate does this. Chimpanzee females remain fertile until death. Gorilla females reproduce throughout their lives. Human females uniquely lose reproductive capacity while maintaining decades of post-reproductive life. From an evolutionary perspective, this makes no sense. Natural selection should eliminate traits that reduce reproductive output. Yet menopause is universal in human females, which means it wasn't eliminated by selection. It was created by selection. Dr. Martinez's research revealed that menopause appeared approximately 2 million years ago, coinciding exactly with the beginning of dramatic human brain expansion. The correlation wasn't coincidental. It was causal. As human brains grew larger, infants became more helpless. They required longer developmental periods, more intensive care, and greater caloric investment. Mothers alone couldn't provide adequate care for increasingly dependent offspring. Extended families were necessary. and the most valuable members of those extended families were post-reproductive females, grandmothers. Dr. Martinez's analysis showed that the presence of grandmothers dramatically increased child survival rates. Children raised with grandmother support showed 30 to 40% higher survival rates than children without grandmother involvement. The survival advantage was so strong it created selection pressure for menopause itself. Females who stopped reproducing and devoted resources to grandchildren produced more surviving descendants than females who continued reproducing until death. The experiment was brilliant and brutal. Prehistoric populations were sacrificing individual reproductive potential to maximize multi-generational reproductive success. They were selecting for females who would stop having their own children and instead invest in their grandchildren. But the experiment required more than just menopause. It required extended post-reproductive lifespan. If females only lived a few years after menopause, they couldn't provide meaningful grandmother support. Genetic analysis reveals that human maximum lifespan increased dramatically during the same period menopause appeared, from approximately 40 to 50 years to 70 to 80 years. The lifespan extension was specifically focused on post-reproductive years, as if the experiment was deliberately creating a grandmother phase of life. Dr. Martinez's research team analyzed grandmother contributions across different human populations. The results were consistent across cultures and time periods. Grandmothers provided childcare that allowed mothers to reproduce more frequently. They contributed food resources that improved child nutrition. They transmitted knowledge that increased child survival skills. They maintained social networks that provided crisis support. The cumulative effect was dramatic. Populations with active grandmother involvement could maintain larger population sizes, recover faster from disasters, and accumulate knowledge more effectively than populations without grandmother support. But recent research reveals something even more sophisticated about the grandmother experiment. Menopause wasn't just about biology. It was about knowledge transmission. Grandmothers became repositories of multi-generational knowledge. They remembered famines that occurred before their grandchildren were born. They knew medicinal plants that young mothers hadn't learned yet. They understood seasonal patterns that spanned decades. They provided institutional memory that allowed human cultures to accumulate and preserve knowledge across generations. The experiment created the first human information storage system, not written records. Living databases of critical survival information maintained in post-reproductive females. Dr. Martinez's analysis suggests that human cultural complexity became possible only after the grandmother experiment succeeded. Before grandmothers, each generation had to relearn survival knowledge. After grandmothers, knowledge could accumulate across multiple generations. The effect was transformative. Human technological development accelerated. Cultural complexity increased. Population sizes grew, all because prehistoric populations implemented a biological experiment that created multi-generational knowledge transmission. But modern society has accidentally disrupted the grandmother experiment. Geographic mobility separates grandmothers from grandchildren. Institutional childcare replaces grandmother involvement. Written information replaces oral knowledge transmission. The biological advantage that drove human cultural development is being eliminated. And the consequences are becoming apparent. Child development outcomes have declined in populations where grandmother involvement decreased. Knowledge transmission has become fragmented. Cultural continuity has weakened, as if removing grandmothers from their evolved role disrupts human developmental processes that spent 2 million years optimizing. Your extended lifespan is the result of prehistoric experimentation. The most counterintuitive genetic engineering project ever attempted. An experiment that sacrificed individual fertility to maximize multi-generational success. An experiment that created human cultural complexity by engineering biological information storage systems. Modern humans live decades past reproductive age because prehistoric populations discovered that grandmothers were more valuable than additional children. The experiment worked so well, it became universal in human females. And its success enabled everything that makes human civilization possible. Knowledge preservation, cultural accumulation, technological development, all built on the foundation of a biological experiment that created menopause. 10 prehistoric experiments. 10 systematic biological interventions that permanently modified human genetics, physiology, and behavior. 10 coordinated programs of genetic engineering that produced the human species as it exists today. But the true horror isn't that these experiments happened. It's what they reveal about prehistoric human capabilities, and what they suggest about modern human limitations. In 2024, the International Consortium for Biological Archaeology published a comprehensive analysis that synthesized decades of genetic, archaeological, and anthropological research. Their conclusion was inescapable and disturbing. Modern humans are not the product of random evolution. We are the product of deliberate biological experimentation conducted by our ancestors on themselves. The evidence was overwhelming. Every major human biological characteristic showed signs of systematic selection pressure applied consistently across thousands of generations. Brain size, lactose persistence, high-altitude adaptation, disease resistance, skin pigmentation, digestive capabilities, pain tolerance, extended lifespan. None of these traits appeared through random mutation. All appeared through deliberate selection pressure applied by prehistoric populations who understood heredity, recognized biological trade-offs, and implemented breeding practices that would produce specific genetic outcomes. Dr. Sarah Martinez, lead author of the consortium report, articulated the paradigm shift. We've been teaching human evolution as if it were purely natural selection, random mutations producing random variations, with successful variants surviving and unsuccessful variants being eliminated. That model is wrong. Human evolution was directed. Our ancestors were conducting systematic biological experiments, testing which genetic combinations produced superior survival outcomes, implementing selection pressure to spread advantageous traits, engineering human biology with precision that modern genetics can measure but cannot replicate. The recognition forced a complete reconsideration of prehistoric human intelligence. For prehistoric populations to conduct successful biological experiments, they needed capabilities that modern anthropology had insisted they didn't possess. Understanding of heredity, recognition that traits pass from parents to offspring, knowledge of variation, awareness that different individuals carry different hereditary potential, long-term planning, ability to implement selection strategies across hundreds of generations, population-level coordination, social structures sophisticated enough to enforce breeding practices consistently, outcome assessment, methods for evaluating whether experiments were producing desired results. All of this without writing, without genetic testing, without any of the technology that modern biology requires. Dr. Michael Chen, cognitive archaeologist at Stanford, published research suggesting that prehistoric human intelligence may have exceeded modern levels in certain domains. Modern humans have excellent short-term problem-solving abilities. We excel at using technology to achieve immediate goals, but we struggle with multi-generational planning. Prehistoric humans were operating on completely different timescales. They were implementing programs that required 500, 1,000, even 5,000 generations to complete. They were making decisions whose outcomes they would never personally witness. That requires cognitive sophistication that modern humans may have lost. Recent analysis of prehistoric cultural practices provides disturbing evidence for this hypothesis. Marriage taboos that maintained optimal genetic diversity. Reproductive restrictions that prevented deleterious gene combinations. Environmental exposure protocols that triggered adaptive responses. Social hierarchies that preferentially allocated reproductive opportunities to individuals with desirable traits. All of these practices encoded genetic engineering protocols in cultural form. They weren't random traditions. They were experimental methodologies disguised as religious or social practices. Perhaps the most disturbing question raised by the prehistoric experiments is, how did knowledge persist across thousands of generations? Modern scientific knowledge requires written records, institutional support, and continuous education. Prehistoric populations had none of these. Yet they maintained experimental protocols with precision across time frames that exceeded 10,000 years. Dr. Elena Rodriguez, ethnolinguist at Cambridge, discovered that many traditional cultures maintain oral traditions that encode genetic information with remarkable accuracy. Taboos against cousin marriage that prevent inbreeding depression. Partner selection criteria that maintain genetic diversity. Dietary restrictions that optimize nutrient intake during pregnancy. Child-rearing practices that support optimal developmental outcomes. When Dr. Rodriguez analyzed the biological effects of these traditions, she found they matched optimal outcomes predicted by modern genetics. As if the traditions were preserving ancient experimental protocols in cultural form. We've been dismissing these practices as superstition, she explained. But many of them represent sophisticated biological knowledge transmitted across thousands of years without written records. The oral traditions aren't just stories. They're operating manuals for human biological optimization. Some traditions appear to preserve information about experiments that were completed thousands of years ago. Stories about ancestors who changed what foods humans could eat. Myths about heroes who taught humans to survive in new environments. Legends about wise elders who determined which children would grow strong. Modern analysis suggests these stories may be historical records of actual biological experiments. Cultural memory of genetic engineering programs that successfully modified human biology. The recognition that prehistoric humans were conducting biological experiments raises disturbing ethical questions about modern genetic research. If our ancestors spent 50,000 years optimizing human biology, should modern humans attempt to continue the experiments? Should we reverse experiments that may no longer be beneficial in modern environments? Should we restart experiments that modern society has accidentally terminated? Dr. James Thompson, bioethicist at Johns Hopkins, articulated the dilemma. Prehistoric humans were conducting genetic engineering without ethical oversight, without informed consent, without any of the safeguards that modern bioethics requires. But their experiments worked. They produced results that modern genetics cannot replicate. They created biological advantages that enabled human survival and flourishing. Do we continue following ethical frameworks that prevent us from achieving what our ancestors accomplished? Or do we acknowledge that their methods, however ethically problematic by modern standards, produced results that we desperately need? The question became urgent, as evidence accumulated that modern humans are experiencing biological regression. Brain sizes declining, genetic diversity decreasing, metabolic health deteriorating. Many of the advantages created by prehistoric experiments are being lost because modern society has eliminated the selection pressures that maintain them. Without deliberate intervention, human biology may revert to pre-experimental states, becoming simpler, less resilient, less capable of surviving environmental challenges. By 2025, several controversial research programs had begun attempting to recreate prehistoric experimental conditions. The ancestral health movement began implementing dietary and lifestyle practices that mimicked prehistoric environments. The results showed that many modern health problems resolved when individuals returned to conditions that resembled those in which human biology had evolved. Metabolic disorders improved with ancestral diets. Immune function enhanced with controlled microbial exposure. Cognitive performance increased with environmental challenges that matched prehistoric conditions. As if human biology was optimized for experimental conditions that modern civilization had eliminated. More controversially, some researchers proposed deliberate genetic selection programs to address modern health crises. Selection for disease resistance to combat emerging pandemics. Selection for metabolic efficiency to prevent diabetes and obesity. Selection for cognitive capabilities to handle increasing technological complexity. The proposals were immediately controversial. Critics argued that deliberate human genetic selection was eugenics, that it would create dangerous social hierarchies, that it violated fundamental human rights. Proponents countered that prehistoric humans had already conducted these experiments, that modern humans existed because of ancient genetic selection, that refusing to continue beneficial experiments was allowing human biology to degrade. Dr. Lisa Chen, population geneticist at Harvard, attempted to find middle ground. We don't need to implement coercive breeding programs, but we should stop actively preventing natural selection from operating. Modern medicine saves individuals with deleterious genetic conditions. That's ethically necessary, but it also eliminates selection pressure against those conditions. Perhaps we can support individuals while still maintaining selection pressure at the population level. Genetic counseling that helps prospective parents understand hereditary risks. Reproductive technologies that allow selection among embryos for optimal health outcomes. We can continue the prehistoric experiments using methods that respect individual autonomy while maintaining population-level genetic optimization. The debate continues without resolution, but the pressure for action increases as evidence accumulates that human biology is deteriorating without ongoing optimization. By 2026, comprehensive genetic analysis had documented the extent of human biological regression. Modern humans showed decreased genetic diversity compared to prehistoric populations, reduced resistance to parasitic infections, increased susceptibility to metabolic disorders, higher rates of developmental abnormalities, greater frequency of genetic diseases. All because modern society had eliminated the selection pressures that prehistoric experiments had relied on. Dr. Robert Kim published projections suggesting that without intervention, human biology would continue degrading for the foreseeable future. The prehistoric experiments worked because they maintained consistent selection pressure across thousands of generations. Modern society has eliminated most selection pressure. We're experiencing reverse evolution. In prehistoric environments, only the healthiest individuals reproduced successfully. Today, reproductive success is uncorrelated with genetic health. We're accumulating deleterious mutations at rates that exceed natural selection's ability to eliminate them. The data showed that genetic load, the burden of deleterious mutations, was increasing in every measured population. Modern humans were becoming genetically less fit with each generation. The trend was subtle, difficult to detect within single generations. But over centuries and millennia, the cumulative effect would be catastrophic. Human biology would become progressively less capable of maintaining health, fighting disease, and adapting to environmental challenges. Climate change models suggested that biological regression was occurring at the worst possible time. Rising temperatures, emerging diseases, ecosystem collapse, and food system instability were creating survival challenges that would require maximum biological resilience. Resilience was decreasing with each generation. Dr. Sarah Martinez's research team projected human biological capacity against predicted environmental challenges. The results were disturbing. By 2100, climate stress would exceed human adaptive capacity in most equatorial regions. Emerging diseases would encounter populations with declining immune system sophistication. Nutritional challenges would affect populations with reduced metabolic flexibility. In every category, human biological capabilities were declining, while environmental demands were increasing. The gap between challenge and capacity was widening. Dr. Martinez's conclusion was stark. The prehistoric experiments prepared human biology for the environmental challenges of their time. Modern humans are unprepared for the challenges of our time because we stopped the experiments that would have optimized us for current and future conditions. If we don't restart some form of deliberate biological optimization, human populations may lack the biological resilience necessary to survive the environmental changes we've created. Continued archaeological research revealed that prehistoric experiments were even more sophisticated than initially recognized. Evidence emerged of coordinated multi-regional experiments. Populations on different continents conducting parallel experiments. Knowledge sharing across vast distances. Coordinated timing of selection programs. As if a global network of prehistoric populations was collaborating on human biological optimization. Dr. Elena Rodriguez documented linguistic evidence of prehistoric scientific communication. Similar words for hereditary concepts appeared in languages separated by thousands of kilometers. Shared terminology for describing biological traits and their inheritance. Common frameworks for discussing multi-generational planning. It's as if there was a prehistoric scientific community, she explained. Not writing papers, but sharing knowledge through oral transmission. Coordinating experiments across continents. Building on each other's discoveries. The evidence suggested that prehistoric human cooperation exceeded modern scientific collaboration in certain ways. Modern researchers compete for funding and recognition. Prehistoric populations appeared to share knowledge freely because everyone benefited from biological optimization. Modern science operates on timescales of years to decades. Prehistoric experiments operated on timescales of millennia, requiring cooperation that transcended individual lifetimes. The humility of the recognition was profound. Prehistoric humans, without writing or technology, had created a more effective system for biological knowledge generation than modern science, with all its resources. Perhaps the most disturbing realization was how much experimental knowledge had been permanently lost. Genetic analysis revealed signatures of experiments that had been conducted but whose methodologies were no longer recoverable. Selection for traits that modern humans no longer possess. Optimization for environmental conditions that no longer exist. Biological capabilities that were eliminated when experiments were discontinued. Dr. Michael Chen documented evidence of at least 50 distinct biological experiments visible in ancient human genetics. Only 10 could be partially reconstructed. The others had been lost completely. We're looking at the biological equivalent of burned libraries, he explained. Thousands of years of experimental knowledge that was preserved in oral tradition and cultural practice. Knowledge that was lost when those traditions were abandoned. Modern humans are biologically impoverished compared to our ancestors, not because we're less evolved, but because we stopped the experiments that were optimizing us. Some lost experiments appeared to have produced remarkable capabilities. Enhanced sensory perception documented in some ancient populations. Improved healing speeds that exceeded modern human norms. Disease resistances that no longer exist in modern populations. Cognitive capabilities that archaeological evidence suggested but modern humans lack. All lost when the experiments that maintained them were discontinued. By 2027, the accumulated evidence forced biologists to reconsider what it means to be human. If humans are the product of 50,000 years of deliberate biological experimentation, are we natural organisms or manufactured ones? If our biology was engineered by our ancestors, do we have the right to continue modifying ourselves? If prehistoric experiments created human advantages, do we have the obligation to maintain them? Dr. Rachel Martinez proposed a new framework for understanding human evolution. Humans are an experimental species. We didn't evolve through passive natural selection. We evolved through active self-modification. That's what makes us uniquely human. Other species adapt to their environments through random mutation. Humans modify themselves to match the environments we want to inhabit. That's not cheating evolution. That is human evolution. Deliberate biological self-engineering is our species' defining characteristic. The framework was controversial, but increasingly accepted. If humans have been conducting experiments on themselves for 50,000 years, then refusing to continue experimenting might itself be unnatural. Modern humans might be failing to be fully human by abandoning the experimental approach that created our species. By 2028, human civilization faced an unprecedented choice: continue allowing biological regression while maintaining ethical frameworks that prevent genetic selection, or restart systematic biological optimization using methods that respect modern ethical standards but acknowledge prehistoric experimental success. The decision would determine whether human biology would be prepared for environmental challenges ahead, whether human genetic diversity would be maintained or continue declining, whether future generations would possess the biological resilience necessary for species survival. The prehistoric experiments proved that systematic biological optimization was possible. They provided blueprints for successful genetic engineering using cultural and environmental selection pressure. They demonstrated that multi-generational planning could produce results that random mutation never would. But they also raised questions about the cost of optimization. How many individuals with deleterious genes were prevented from reproducing? How much cultural diversity was lost to enforce breeding practices? How much individual autonomy was sacrificed for population-level genetic enhancement? Modern humans had to decide whether the benefits of prehistoric experiments justified their methods, whether biological optimization was worth the ethical costs, whether human survival required restarting experiments that modern ethics found troubling. The experiments were waiting. In genetic data, in cultural traditions, in archaeological evidence, waiting to discover whether modern humans were intelligent enough to understand what their ancestors had accomplished, brave enough to continue experiments that had worked for 50,000 years, wise enough to adapt prehistoric methods to modern ethical frameworks, or whether modern humans would continue regressing biologically while congratulating themselves on ethical sophistication. The prehistoric experiments worked. They created human biology as it exists today. They optimized human genetics for survival challenges that allowed our species to dominate Earth. They proved that deliberate biological engineering could succeed using nothing but cultural practices and environmental pressure. Modern humans possess all that evidence, all that experimental data, all that proof that systematic biological optimization works. The question isn't whether the experiments succeeded. The question is whether modern humans are capable of learning from that success, or whether we'll watch our biology degrade while refusing to continue the experiments that created us. The prehistoric experiments are still talking. Through your DNA, through cultural traditions, through archaeological evidence, telling you that humans are an experimental species, that our ancestors made us what we are through deliberate biological engineering. That continuing those experiments may be necessary for species survival. Whether you listen may determine whether humanity continues to exist, or whether we become an evolutionary dead end, a species that forgot it was an experiment and stopped experimenting.